All right, I think we'll go ahead and get started. So welcome everyone. This is second year of our Rad Genomics event. Professor Vicky will be joining me in about an hour or so, but really excited to see everyone here. We have a really great lineup of speakers, including our opening keynote, and we have two great keynote speakers for today, and then we have a mixture of talks to really build off momentum from last year. So I'm excited. This is for those who are unfamiliar with the event. Ken Kennedy Institute has been running a conference series specific to AI in healthcare and related to genomics as well for the last few years. And last year we decided to do a pre-conference event or workshop. And again, given the response and the feedback, we decided to go for a second year, which is really exciting. And just to introduce myself, I'm Todd Trangan. I'm an associate professor in the computer science department here at Rice. I've been here, this is my 6th year at Rice, and again, excited to kick things off. In terms of logistics, I think our registrations are about 10% higher than last year, which is always good to see. So getting some kind of sign of interest and momentum and encouragement to run this event again. One day I hope we can fill up this entire room. Maybe we're little by little working towards that. So it's a really nice venue, but again, just given the increase in registrations, that's always a positive sign. So I'll just go over a few slides. We're starting a little bit late. I wanted to give everyone a little bit extra time for morning traffic to get in, but our schedule is pretty relaxed, as you'll see. We'll have a mixture of breaks. We have a panel discussion in the middle of the day. So really, this is meant not only to present some research and discussion around the topic of genomics, it's also meant as an opportunity for all of us to kind of interact and engage throughout the day. And again, the schedule is pretty relaxed, several breaks and lunch, and then we have a social at the end of the day. So just first, I always like to start with acknowledgements here and just acknowledge the Ken Kennedy Institute team. You'll see them referred to as K2I. Really fabulous team that's supported not only this event previously and now, but several events that I've organized or co-organized. So thankful to Michelle, who you've seen here previously. She was just in the room. And Kelly, in addition to Sammy, who's at the registration desk, to Executive Director Angela Wilkins, and the Director, Lydia Kavraki. So really exceptional team, exciting to have their support to run these type of events. But then, again, as a huge highlight of today is we're kind of kicking off and ending with two fabulous speakers. One is Dr. Fritz Selacek, who's here, and I'll introduce him in a second. Really excited to hear his talk, and I think it really will help us set the stage for what this event is about. And again, I'll get into it in a second, but really just keep in mind it's about reading, assembling, analyzing, and designing genomes, and each of those words are kind of just to make the acronym work, but also there are other obviously implications of those terms, and we had a really, again, great inaugural event last year, and we're excited to kind of continue the discussion. And then as our second keynote, Dr. Stephanie Hicks, really excited to meet her and have her give her talk later today. So again, really excited about the speakers, and those are our keynotes, but we have additional invited speakers that complement out the topics. And so my goal is by the end of today, you'll have heard about topics in all 4 categories and had a chance to participate in discussion and interact with the speakers and attendees. And of course, all the attendees. These events, even though we've worked hard to make it very affordable and if not free for everyone to attend, and thanks to the support of K2I, everyone obviously realize everyone has a busy schedule, and there are additional events in the week as well. So appreciate your time commitment today for this event. So just again, for those that did not attend last year, just to give a highlight what this is about. So usually I start out an event by thinking of an acronym and then fitting topics inside of that. So this one actually was the other way around. We really wanted to think of genomics through these 4 topics. You say reading, but really sequencing, and we'll hear some of this through the first keynote. But really, there's been tremendous advance over the last decade and longer, of course, but especially in the last decade. I started getting heavily involved around and kind of thinking about sequencing around 2010, 11, it's been amazing to see the advances since then. And you'll hear more about that from a leading expert in this topic, but really our ability to read longer stretches of DNA and how that's really changed a lot of the downstream applications and results we can get out. But once we read or sequence a genome, we then have to think about assembling it often. And again, you'll hear some of this from the first speaker, but there's been a tremendous amount of advance, not only in methods for assembling genomes, but actually high quality assembled genomes. So those first two topics really set the stage for kind of what we can do afterwards, specifically with genomics focus. And then the second session is really thinking about analyzing and designing two very broad topics, I'd say, but analyzing it specifically is thinking about what now we have telomere to telomere assembled genomes. What do we do with them now? What can we use them for? What's the advantage of having them? Others have obviously eloquently made the case and what we could do with this, but we'll kind of revisit some of that specifically with respect to interpreting and integrating this information into resolving biological phenomena that perhaps are poorly understood or maybe helping prevent or cure disease. Really, really exciting topics that we can start thinking about now. And then designing genomes, really just thinking about the advances in bioengineering and synthetic biology, where we can really start thinking of it as we understand genomics more, we can actually start using this to our advantage to help treat disease or look at environmental contamination, et cetera. There's a lot of really exciting applications in using kind of the idea of like designing, okay? And last year we had a really rich discussion on this topic, and I hope we can talk through this again during the panel discussion. But that's really to set the stage to kind of give you all a little bit more background on why we're here today. Okay, so just summarizing, really why organize this workshop again, and so a primer on the topics I just covered for, I'm assuming no background knowledge but from any audience member. You may know a lot about these topics, you may not know very much about this. That's all good, that's the design of the workshop. We really want to encourage broad engagement across Houston, with Texas Medical Center, at Rice, at other universities in the Houston area, and outside, even in the Gulf Coast region here. But really, there's no assumed knowledge. If all of these terms are new to you, that's great, welcome. And if a lot of this leads into your expertise, that's also great, because I think that's going to promote great discussion. So that's topic two. And then just the third thing is really just trying to promote collaboration. This may serve as an opportunity for perhaps you've been interested in collaborating with someone on some of these topics, and you can reach out and meet them today and maybe follow up with them. The other comment is all of the talks today and materials should be online and available via recorded material. So if you want to share this with anyone after you attend the event, if you think it was useful, feel free to do so. That's one of, again, a nice aspect of partnering with K2I. They provide a lot of that organization and facilitation of sharing out the content once we've created it here. But yeah, just revisiting the schedule. This is a high-level schedule, so we're running a few minutes late, but that's all okay. The schedule is kind of designed to be a little bit flexible. And so just high level, I just want to emphasize we kind of have two sessions, opening keynote, closing keynote, the kickoff and close, and a panel discussion in the middle of the day. And we were thinking about what to do with this time, but last year's panel discussion I felt was very productive and useful, so we decided to go revisit that with a few tweaks here. And so specifically the morning session, I'm gonna, as soon as I stop talking, which will be in just a minute here, I'm gonna hand it over to our first keynote speaker. We'll follow by maybe a 15-minute break or so, and then we'll have two invited talks, which I'm excited to have.